Pesticides are chemical or biological agents which are used for eliminating pests. We have three different categories of pesticides, such as organochlorine, organophosphate, and carbamates. Again, these three different categories are divided into two different types, such as persistent and non-persistent insecticide. When we talk of persistent and non-persistent insecticide, we usually refer to the capacity of a particular pesticide, whether it survives in environment for a long time or not. If it survives for a long time, once it is utilized for a desired purpose in the agriculture sector, then we call such pesticides as persistent insecticides, because they are quite stable and they show relatively less solubility in water. They are less volatile. On account of these properties, such insecticides survive in environment for a very long time. As far as non-persistent, so they are relatively unstable and they do not survive because they degrade automatically once they are utilized for desired purposes. As far as persistent insecticides are concerned, or organochlorine, we have an example of DDT. Here we have drawn a structure of DDT which is known as dichloro-diphenyl trichloroethane. We have two carbon atom chain, three chlorine atom are attached on one carbon atom and another carbon atom holding hydrogen, and two similar group which has a phenyl ring to which a chlorine atom are attached to a para position. So the name for this molecule is dichloro-diphenyl trichloroethane. We will focus on non-persistent insecticides. Basically, they are powerful neurotoxins. Neurotoxins are those chemical or biological agents which act upon a nervous system for paralyzing organism. So they are very toxic as compared to organochlorines. Their action is quite potent and they act very swiftly as compared to persistent insecticides. For example, organophosphates are very dangerous. They usually fall in the category where nerve agents belong to, such as serine, VXR, mustard gas. These belong to a nerve agent family. These chemical agents have used in the past for paralyzing the enemy's soldiers. But before we know the action of non-persistent pesticides such as organophosphate and carbamate, we must briefly focus on the basis of a nervous system. So nervous system is made up of individual cell known as neurons. Here we have a neuron. The structure I have drawn consists of a cell body, dendrite, which are cytoplasmic extension. Here the nerve impulse is taken up by the nerve cell and it transfers along the filament coated by myelin sheath and ultimately transmit into another neuron via hairy filament. But two neurons are linked by a microscopic gap known as synapse, and it is this synapse where a neurotransmitter molecule release that act as a bridge for transferring a nerve impulse from one neuron into another neuron. Actually, what happens in a healthy prey or healthy insect, they have an enzyme known as cholinesterase. These enzymes usually hydrolyze the neurotransmitter acetylcholine. So this acetylcholine is a sort of neurotransmitter which release into the synapse for transmitting nerve impulse between two neurons. Once the neuron is paused or it need to be stopped, so these acetylcholine molecules are hydrolyzed or they are destroyed by this enzyme cholinesterase. So by this way the transmission is stopped and the coordination of the nervous system remains stable. But if we use carbamate or organophosphate, what they do, actually they destroy this enzyme, this cholinesterase enzyme. For example, here we have normal reaction that take place in a healthy. In fact, this is, let's suppose, EOH is a representation for cholinesterase enzyme. It reacts with the acetylcholine molecule in a nucleophilic addition reaction, and we get acetyl enzyme. Here we have acetyl enzyme, and a choline molecule is released, and this acetyl enzyme is ultimately hydrolyzed by the water for giving acetic acid, and the enzyme is recovered again. It is recovered again for another action. So this is how the acetylcholine molecule is neutralized for stopping the flow of nerve impulse. But if no, we consider two different cases here, case number 1 and case number 2. So in case number, in both cases we have replaced acetylcholine molecule with these non-persistent insecticides. Here we have such as organophosphate, pesticide. It captures the enzyme after release of HX molecule for giving a phosphoryl enzyme. In the second case, a carbamate pesticide, this is a carbamate pesticide, which again captures the healthy cholinesterase enzyme. After release of alcohol molecule, it gives carbamyl enzyme. Now, whether carbamyl enzyme or phosphoryl enzyme, they are not very much soluble in water, so they do not react with water for releasing the enzyme so that it could stop the acetylcholine. Once this enzyme is captured by both the type of pesticide, so the acetylcholine will continue to pour into the synapse, and it will trigger the unstoppable flow of nerve impulse. So this unstoppable flow of nerve impulse ultimately paralyze the prey and kill it. So this is all about the non-persistent insecticides that are very powerful neurotoxins.